Hello, students. So, in the reproduction in animals chapter, in this lecture, we will discuss regarding the types of asexual reproduction. A lot of types we have already discussed, and one or two left. So, that we'll discuss. After that, we'll go for the human sexual reproduction. So, here, the use of plant tissue culture that we were discussing in case of our in case of our last lecture. So I can tell you one thing: the use of plant tissue culture we are using for different purposes, like just a minute. Let me take the pointer first. First thing is the to get the higher yield. Yield means number of crops will be higher, and independent of environmental environment or season, then potential for low cost production, then harvest in short duration, production of novel compounds round the year availability. That means the crop should not be season dependent, so there will be no scarcity, and continuous supply of products with uniform quantity and conservation of natural population and sustainability. Okay, so these are the different reasons why we are using the plant tissue culture. Now, the method offers the advantage over other methods is that disease free, we can get disease free plants from this tissue culture, and new plantlets by the score of thousands. It has been used extensively for the production of commercially important plants like tomato, banana, apple. So there are several things. That means the requirement for which crops or which fruits are very huge. At that time, we are going to use the plant tissue culture. And here also in the orchids also we are using the plant tissue culture. The most notable example of the application of micro propagation, that is the farming of orchids. You can see the orchids flower. So that farming of the orchids, that we are using the plant tissue culture. Now, spore formation. This is another method of the asexual reproduction. So here in the spore formation, you have the spores here, you have the spores here, you have the spores here. So this spore formation is another means of asexual reproduction. And during unfavorable conditions, the organism develops sac-like structure called the sporangium, and this is the sporangium. Okay, so this is sac-like structure. You have the sporangium and it contains that sporangium, it contains spores. It contains spores. And when the conditions are favorable, the sporangium burst opens and spores release germination to give rise to new organisms. So here you can see that the food source is this one. This one is the food source, and this is our hyphae. And these spores at that time, whenever this sporangium burst opens, then spores are released and germinate to give the rise of new organism. Rise of new organism. Rise of new organism. Okay. Next, so sporangium. So spore formation, this is another asexual reproduction. Now advantage of the asexual reproduction. First is mates are not required because single parent cell is involved here. The process of reproduction is rapid. So shorter time period we require. Shorter time period we require. An enormous number of organisms can be produced in very less time, and positive genetic influences pass on to the successive generation. Positive genetic influences pass on to the successive generations, and it occurs in various environments. It occurs in various environments. Now, the disadvantage of asexual reproduction, lack of diversity, most identical we are getting. So there is no differences between the daughter cell and the parent cell. If one cell is affected by the diseases, then all the batch will be suffering from the disease. Some nutrient deficiencies are there in one cell, then all other cells will get the same thing. So negative mutation persists for generation by generation, and only one organism is involved. Diversity among the organism is limited. They are unable to adapt the changing environment, and a single change in environment would eliminate the whole entire species. These are the disadvantage of the asexual reproduction. Now, asexual reproduction examples, we know that bacterium undergoes asexual reproduction, blackworms, mudworms, hydras, organisms such as copperheads undergo the parthenogenesis, and sugarcane that can be grown with the vegetative propagation. Now, the difference between sexual and asexual, this is very, very important. This is very, very important question. So, please write down this one. Two parents take part in sexual reproduction, but asexual, single parent will take. And variation occurs in offspring. And here, offspring are genetically identical to each other and to their parent. Fertilization takes place. Here, no internal fertilization or external fertilization is there. Gametes are involved in sexual reproduction where no gametes involved. Mixing of hereditary material is there. No mixing of hereditary material is there in asexual reproduction. In asexual reproduction. So, now few questions may come like this. What is budding? First, let me change the color of the ink color. Okay. Answers. A form of asexual reproduction in which a new individual develops from the some generative anatomical point of parent organism. This is called the budding. So, a form of asexual reproduction in which a new individual develops from some generative anatomical point of the parent organism. What is Hydra? Genus of invertebrate freshwater animals of the class of Hydrozoa. That is called the Hydra. So here you can write down genus of invertebrate freshwater animals of the class Hydrozoa is called a hydra. Next, what is an embryo? An unborn human or animal in the earliest stages of growth when its basic structure are being formed is called the embryo. So, an unborn human or animal in the earliest stages of growth when its basic structures are being formed is called as an embryo. Now, what is internal budding? Internal budding, that is an internal budding is an asexual reproduction process favored by parasites and favored by parasites. It is a process unique process in which two daughter cells are produced inside a mother cell, which is then imbibed by the offspring prior to their separation. Now, what is exogenous budding? Exogenous budding is a type, just a minute, is a type of, just a minute, is a type of asexual, is a type of asexual reproduction displayed by organisms such as Hydra. A new organism develops as an outgrowth or bud on the surface of the mother cell during this process, and it expresses itself extremely on the parent. It expresses itself extremely on the parent. Okay. Now the sexual reproduction. Let me change the pointer color. Okay. Sexual reproduction, it may be internal or it may be our external fertilization. Internal fertilization and external fertilization. Internal fertilization and external fertilization. So here you can see that the human and dogs, they are doing the internal fertilization because that will be inside the body, and external fertilization like reptiles and birds, and they are doing the external fertilization. So self-fertilization. Here, certain invertebrates that such as tapeworms, they are certain other parasites also. They are reproducing through the process self-fertilization. What happens in this? They fertilize their eggs with their own sperm. So they fertilize their eggs with their own sperm. So that is the self fertilization. Now sexual reproduction in humans. Now first is the male reproductive system, because you know that the sexual reproduction in humans, that will be done by the male and female also. Female egg and male sperm, at that time that will fuse, and at that time that will create the zygote. Okay, so male reproductive system, with the main thing is the pair of testes, a pair of sperm ducts, and a penis. And the testes are located outside the male body. The testes are located outside the male body. Here you can see this is the testis, this is the testicle, testes, and sperms are produced in the testes, and sperms are the male gametes here. And this one is the penis, this one is the urethra, and this one is the vas deferens, and this one is our seminal vesicle. This is our seminal vesicle. Okay. And testosterone, this is the hormone. Okay, this is the hormone. This is the hormone, and this is the male sex hormone, and it's produced in the testes. Okay, testicles. And testosterone, that is responsible for the development of sex organs, production of sperms, and also development of secondary sexual characteristics. Here you can see this is the male gametes, where you have the head, nucleus, and mitochondria. This is the head portion, this is the mitochondria portion, and this is the tail portion. Okay, so this one is the sperm. And testosterone, this is the hormone, that is primary hormone, male sex hormone, and is produced by the testes. Now the sperms here, you can see that this is the male gametes, which is going to be reproduced. This is responsible for the reproduction of the human zygote. And here you can see that sperms are the male gametes produced by the testes. These are the male gametes. So these are the male gametes. Okay, and millions of sperms are produced by the testes in a day, and each sperm is a single microscopic cell that having a head, having a middle piece, that is mitochondria, and there is a tail. So there is a head, middle piece, and tail, and each sperm, that is a single microscopic cell. Now head contains a nucleus. Here you can see that this is a nucleus. Head contains a nucleus with single set of chromosomes. And the middle portion, the middle portion, that piece contains a large number of mitochondria, and that provide the energy during the active movement. And sperm is a motile gamete that which moves through the female reproductive tract with the help of the tail. One thing I have told you, female egg, that is immobile, but here the sperm is the motile gamete, and it will move through the female reproductive tract with the help of this tail. Now the female reproductive system. Here you can see that for the female reproductive system, this is the vagina. This one is the cervix. This one is the endometrium. This is the ovary. This one is the fimbriae. This one is the fallopian tube. And here, this is also fallopian tube, and this is the uterus. Okay, so the female reproductive system consists of pair of ovaries. Here you can see that this is one ovary, this is another ovary. Just a minute, let me change the pointer colors. Okay, this is one ovary, this is another ovary, and pair of fallopian tube here. This one is the fallopian tube, this one is the fallopian tube. So what we have, pair of ovaries and pair of fallopian tube. A single uterus we have here, single uterus, and ova are produced by the ovaries. So here you have the ovary. These ovaries, this will produce the ova. And fallopian tubes, that is oviducts. These fallopian tubes are the oviducts that carry the ova from ovaries to the uterus. Okay, from these ovaries to the uterus, towards this uterus, that ova will be carried out. Okay, and uterus that holds the growing embryo, and thus helps in holding the pregnancy. So in the uterus, actually the small baby that will be there. Okay, so in the uterus the embryo will be there, and after the maturity you can see that there will be it will hold the pregnancy for 38 weeks. Now the estrogen or estrogen. So estrogen or estrogen, that is the, let me change the in color first. Okay. So estrogen, that is the primary female sex hormone secreted by the ovaries, and it is responsible for the development of female reproductive organs and secondary sexual characteristics, development of female gamete, ovum, and maintenance of menstrual cycle. Maintenance of menstrual cycle. Now the ovum. Here you can see the human egg, that is the ovum. Now, the ovary produces the female gametes. The ovary produces the female gametes, where you have seen that this was the ovary and through which the fallopian tube has been connected here, and from here also the fallopian tubes connected, and this is the ovary. So, the ovary produces the female gametes are known as ovum or ova. Okay. So, in human being, a single mature egg or ovum is released into the oviduct and fallopian tube by the respective side ovary every month. And the ovum is a single cell and is each such larger than the sperm, and it is non-motile. It is non-motile, immobile, non-motile, and is carried to the uterus by the oviduct. By the oviduct. Here you can see that. Just a minute, the figure. Here you can see that from there the ova will be created, and after that it will move to the uterus and through the oviduct. Okay. So this is the human egg where you have the nucleus, and here you can see the cytoplasm. These are the cytoplasm. These are the cytoplasm, and this is the corona radiata, and this is the zona. This is the zona pellucida, and this one is the first polar body. So this is the ovum. Now the fertilization. So fertilization, that is the process where the male gametes and the female egg. So actually that sperms, they are coming towards the egg, and after that the zygote will be produced. So male gamete with the female gamete, fertilization means this is the process of fusion of the male gamete with the female gametes, right? So here you can see this is the fallopian tube, this is the fallopian tube, this is the ovary, and these from these you can see the ovary. You can see that in this uterus, this is vagina, this is vagina. In this uterus, you can see that this is uterus. In this uterus, the sperm will be acting on the egg, and in this uterus, you have this type of the sperms are coming, and this is the egg. This is egg is immotile, and these are motile. Now this will react fusion. There will be fusion. After fusion, there are two cells, then four cells, then eight cells. Like that, the zygote will be created, and you can see that after few weeks there will be a child or offspring. Okay. So here you can see the changes. This is the day 0, and day 1 first cleavage, then day 2 there will be 2 cell stage, day 3 and 4 cell stage, then 8 cell uncompacted morula, then 8 cell compacted morula will be there, and early blastocyst you have day 5, day 6 7 you have late stage blastocyst, and then 8 9 implantation of the blastocyst. So here both the gametes containing haploid set of the chromosomes fuse and thus maintain the diploid double number chromosomes here. So here you can see that 23 and 23 from the mother and father, then total 46 chromosomes will be there. The fertilized ovum, that is now called as the zygote. Now, in human, fertilization takes place in the fallopian tube. This is the fallopian tube. This one is the fallopian tube. So, this process, the fertilization process will take place in the fallopian tube, and then zygote is carried to the uterus. So, this one is the uterus. This one. Okay. This one is the uterus, and inside the uterus it undergoes repeated divisions and grows and becomes embryo. and becomes embryo. Now, steps of fertilization in humans. Here, acrosomal reaction. So the sperms in capacitation undergo acrosomal reactions and release certain chemicals known as sperm lysines and present in the acrosome. And due to acrosomal reaction, the plasma membrane of the secondary oocyte and sperm are fused together so that the contents of sperm can enter into inside the female reproductive system. Okay. So when plasma membrane of the sperm binds that with that of the secondary oocyte, the plasma membrane of the oocyte depolarizes, and this prevents polyspermy. This prevents polyspermy. And calcium ions that play a significant role in acrosomal reaction. And the main factors essential for acrosomal reaction are optimum pH, the temperature, calcium and magnesium concentration. So these are required. Now the cortical reaction. Now whenever the fusion of the plasma membrane will happen, then oocyte shows cortical reaction. Cortical granules found under the plasma membrane of oocyte and which fuses with the plasma membrane and releases cortical enzymes between the zone pellucid and plasma membrane, and the zone pellucid is hardened by the cortical enzymes and prevent the polyspermy. Now sperm entry, this is the projection known as the cone of reception, is formed in the secondary oocyte at the point of sperm contact, and this cone of reception receives the sperm. Then karyogamy, here karyogamy, after the entry of sperm, suspended second meiotic division is completed. So here meiotic division, here meiotic division will be there, is completed by the same secondary oocyte, and this gives rise to the haploid ovum and the second polar body. So the head of that sperm, we have seen that sperm is like this one, and this head of the, and there is a tail also. So this is the middle portion and this is the head of this sperm where you have the nucleus, and this is the tail portion. So here the head of the sperm containing the nucleus that detaches from the entire sperm and is known as male pronucleus. So this nucleus will be detached and it is known as male pronucleus. Male pronucleus. And the tail and the second polar body degenerates. The nucleus of the ovum is known as female pronuclei. The nucleus of the ovum, that is known as pronuclei. Pronuclei. Then male and female pronuclei fuse and their nuclear membrane degenerate. So fusion of the chromosomes of male, 23 chromosomes from male and 23 chromosomes from female gametes is called the fusion. The fusion process, this is called the karyogamy. Karyogamy. And the ovum is now fertilized and is known as zygote. And now the zygote will be formed. Activation of the eggs, here after the entry of the sperm triggers the metabolism of the zygote and protein synthesis will be there and cellular respiration that will increase. Now implantation, once the fertilization happens, the cell starts to divide and multiply within 24 hours in the fallopian tube. So you have seen the picture that first it will be single one, then it will be two, then it will be 4, then it will be compact, then it will be compact here, then it will be compact here. So this detached multi-cell structure is called the zygote, and after 3 to 4 days it travels to the uterus. And now we can call, whenever it will be inside the uterus, whenever it will be inside the uterus, we can call this is our embryo. This is our embryo. And the embryo develops and undergoes various stages and gets attached to the endometrial layer of the uterus. So this process attachment is called the implantation. Now sex determination, the fertilization, that is the process. Now sex determination, the female that has only the XX chromosome, the male that has the XY chromosome. If this X and this Y that will be created, then it will be our, if the sperm carries a Y chromosome and the child will be a boy, and if sperm carries the X chromosome, at that time the child will be a girl. So boy, if this will be XX, then this will be a girl. Okay. So the total thing that will be dependent on the sperm. With that sperm carries X or Y. Okay. Now few questions are there. So let me try to answer all these questions. Before that, I just want to take the ink color change. Okay, what is the human fertilization? So the male, sorry, the fusion of the male and female, the fusion of male and female gametes occurring in the ampulla of the fallopian tube is known as fertilization 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 in human. Fertilization in human. Next, how long does it take for the egg to get fertilized? So the egg is alive for about 12 to 24 hours after its release from the ovary. And if it is not fertilized by this time, it disintegrates and is shed off by the uterine lining. How long does sperm live? The sperm stays alive for about 72 hours. The sperm swim upward to the fallopian tube to fertilize the egg. The faster that sperm swims, the earlier it reaches the egg. And what are the phases of fertilization? The different phases are, I'm only writing the phases. Penetration, where that sperm releases actosomal enzymes to penetrate inside the egg. Then activation, here then we have the activation. Activation means where the egg membrane depolarize and fusion of the nuclei and formation of zygote. What is the sole purpose of fertilization? The different phases of the fertilization include that penetration, already we told. Now what is the main sole purpose? The purpose of fertilization is to combine. The purpose of fertilization is to combine the haploid chromosome sets haploid chromosome sets of two individuals into a single diploid cell known as the zygote, which is the result of activation of the egg. Now, what are the different types of fertilization? There are two types of fertilization. One is the internal fertilization and another is the external fertilization. External fertilization. What would happen if fertilization does not occur? If fertilization does not occur, that means there will be no zygote formation and egg will degenerate and shed the uterine lining. What is implantation? It is the stage where the embryo adheres to the wall of the uterus. Okay. So this much for this chapter. Thank you all. Thank you.